Good morning, everyone. We are here today to make an interview in relation to Project Protos. First of all, I will ask the PI, Professor Juan Manuel García Ruiz, to give a 10 minutes presentation of the group and the proposal. And then the panelists can make all the questions that they consider necessary. So please, Professor, you may start your presentation now.
Okay, good morning, everybody. Thank you very much for selecting our Synergy Project product for this interview. Our planet is 4.6 billion years old. Every centimeter in this stick represents 100 million years in the history of our planet. We call the first 500 million years, this part in red, the Hadean, because it was thought until recently that the surface of the planet at that time was a hell, like in this picture. So it was covered by river of lava, by volcanoes, bombarded by meteorites. So a landscape in which the probability for life to emerge was zero. We have no rock from that time, no rock at all, except some very tiny crystal of silicon who give us a very important information for understanding our project. This crystal of silicon tells us that 4.4 billion years ago, so very early, just after the formation of the moon, there was liquid water on the surface of the planet. This information is essential because this allows us to reason a completely new view of the Hadean as a cradle for life. So when water started to condense 4.4 billion years ago on the surface of the planet, they do on a crust mainly made of iron and magnesium silicate. And it is very well known that when water interacts with this mineral, trigger a reaction called serpentinization. This reaction releases molecular hydrogen that upon reaction with CO2 will create an atmosphere rich in methane, an ideal organic synthesis laboratory at planetary scale. In addition, this reaction also releases high pH water, so that the sea at that time were alkaline, soda lakes. And the water of this lake, or this sea, in interaction with the granitic rocks that contain the crystal, the silicon crystal I mentioned you before, will enrich the water in silica because the solubility of silica increase exponentially with pH. So this huge mobilization of silica will explain why all the oldest rock in this planet, what we call the Archean rocks in blue here in this stick, all them are silicified. So silica was everywhere in the early Earth, everywhere. And we claim that this molecule, silica, play a key role in shaping the geochemical landscape in which life arose and flourished. But it's not just a matter of ubiquity. There is much more about silica. The first thing is that silica triggers prebiotic reaction. I'm sure that you know the famous lining experiment performed by Stanley Miller, in which he demonstrated the synthesis of amino acids from a reducing atmosphere. We are revisiting this experiment and demonstrate that the borosilicate reactor used by Stanley Miller and all the people working in this area later affect dramatically the gelding of the experiment. In other words, if you made the experiment, a similar experiment with an inert Teflon reactor, the experiment doesn't work. More than that, the brown organic matter forming in this experiment, we show that they self-organize to form vesicles, to form microstructure, biomimetic microstructure, which are hollow. So it means that both the molecular brick of light and the microstructure, biomimetic microstructure, hollowed, able to localize this structure to trigger further polymerization, form simultaneously from scratch. And these have enormous importance for the understanding of how the transition from pure inorganic chemistry, pure geochemistry, to biochemistry takes take place for the origin of life. It is also known that silica at high pH induce the self-organization of minerals. What you see in this slide are not microorganisms. They are not living beings. They are just pure inorganic structure, self-organized by the action of silica. On your left side, carbonate biomorph. On your right side, what we call metal oxide osmotic structure, also called a biomorph. And it has been demonstrated that carbonate biomorph are able to absorb biological macromolecules. And in the case of the osmotic structure, it has been also demonstrated that the membrane forming this biomorph are able to catalyze specifically the condensation of formamide, catalyzing the condensation of amino acid on one side of the membrane and the condensation of nucleobasic on the other side of the membrane. So we plan to explore the fundamental of this fascinating phenomenon, and we will plan to do it within the atmospheric condition we predict for the Hadean. So we will perform this lining experiment using the condition in which we predict for the initial time in the Hadean, methane-rich atmosphere, and then later on we move toward carbon monoxide and CO2-rich atmosphere. So our idea is to explore this fascinating phenomenon, looking for the plausibility of the role in the extending the mineral self-organization, and how this could help to the transition to biochemistry. We performed this experiment in my laboratory of crystallographic studies in Granada, and will be performed by my team, but also for other members of other teams that will join us. All the microorganisms, the first microorganisms arose during the Archean times, during the first billion years of the history of the planet. Identify the renan is a very controversial task. Do you know why? Because all of them are silicified, all of them fossilized in chairs, in silica. So we face a problem of light detection, a very important problem of light detection. How to distinguish unambiguously the renan of genuine microorganisms from non-biological, organic, or silica biomorph. So we had to face this problem, which is key for set the timing of life in the solar system. And this is why we have Dr. Marvin in our team. Mark is an international expert in geobiology, in Archean geochemistry, in life detection, and he will study in breast the preservation of different kind of organisms. So we will use actual microorganisms, we will use organic compound, prebiotic, non-biological organic compound, we will use non-biological silica and organic biomorph, and we will study the silicification of all these structures and compare them. More importantly, Mark and the team will do the experiment upon increasing temperature and pressure. So as you can see, it is obvious that silica is key for our project. So it's something which is obviously extremely important. But surprisingly, the solution chemistry of silica is poorly understood. And this is a big problem for us, because in order to decode the information contained in the Archean rocks, in the silicified Archean rocks, we need a much better understanding of the solubility of silica, of its chemical speciation, of the organization at molecular level, on the organization to have different pre-nucleation clusters, and then polymerization to set the formation of gels. And in the field of chemical precipitation, there was a big move in the last 15 years, and one of the scientists behind this innovation is Professor Helmut Hoffen. Helmut is joined the group to apply the same technology that he already applied for the study of the pre-nucleation class of calcium carbonate and calcium sulfate to the problem of silica. So he will use the same technology and new technology we are jointly designing in order to study in detail the solubility and the precipitation of silica, with the main focus at molecular organization and in the formation of silica, carbosilica nanostructure that we anticipate are very important for our project. In the field of original life, everybody is looking for bizarre, for original, for magic chemical reaction. We are too. But for us, it's extremely important, and now we think that there is nonsense to make a study of original life outside a proper geochemical framework. And this is why we have Professor Wolfgang Bach in our study. Wolfgang is an international expert in the field of rock fluid interaction, with a specialization in the problem of serpentinization, and he is leading a laboratory in the University of Bremen, which is perfectly suited to study the rock. I mean, the whole rocks affect and control the composition of fluid, including the activity of iron and silica. And he will do not only from thermodynamical point of view, but also using kinetic approaches. So using an array of laboratories that we call the HDN simulator. So it's an array of laboratories that are located in different institutions, but that we will use by the same people. Okay, we plan to merge information obtained at the atomic and nanometric scale with petrological, geochemical, geological, biological information up to planetary scale. So we, with this synergetic approach, we feel that we will have obtained the best toolkit that we can use in order to understand the information encoded in the silicified Archean rocks, to identify unambiguously all the signature of life here and elsewhere, to explore the limits of mineral self-organization, and then also to see how this mineral self-organization move toward prebiotic chemistry and later on to the original life. Thank you very much for your attention.
Okay, thank you very much for your presentation. Now, each of the panel members will start to make a series of questions that may be answered by any member of your group. We will start with Dr. Fermín Otalora, who can start making the questions now.
Hello, good morning. Interesting talk. And I have some questions. First, you know we can only fund 30 projects. So why is your topic one of the 30 most urgent questions in European science at the moment?
Yes, I'll be happy to answer. Actually, at the moment, the origin of life and the tracing of life is a very hot topic worldwide. There's a lot of effort being made right now to search for life traces on Mars. So there's actually several efforts by space agencies to also better understand how life actually originated early on Earth. So right now is really the time of great interest internationally for this type of research. And given our model for a worldwide process for the origin of life that is triggered by Silica, we feel that our project is very timely at this moment.
Okay. And what will you do if we decide not to fund this project?
We'll continue doing the work we are doing and focusing in on silica as a key compound. The work we've been doing so far firmly points towards a key role of silica in early Earth environments. If you don't fund us, we'll do do what we can to continue the preliminary type work we've been doing. But of course, we need the funding to push the envelope and really test the role of silica. So we hope the non-funding scenario will not materialize.
Okay, and on the other side, being optimistic, if you get the project, what do you expect your first major joint result to be?
Well, I cannot hear. Yes.
What do you expect your first major joint result to be?
Oh, see, well, you know, in the first 2 years, we have several experiments that are facing very important problems, but I will say that I will be very happy. I think that we all us will be very happy if we are able to demonstrate the formation of silica carbon microstructure and how they will interact and impact of the diversity, molecular diversity of the experiment on prebiotic chemistry experiment.
Okay. Which are the main questions you address and why? Not the set of questions in figure B1, 2, but just the 3 top level questions.
So yeah, we have actually several key questions, and this is basically how did the transformation occur from the abiologic world to the biologic world? First is the step towards prebiotic chemistry, then the step to the actual origin of life, and finally the preservation of life. And the key here is the role of silica. But silica is the sort of connecting factor here between these 3 steps towards life and its preservation.
Okay, I do like the schematics in figure B2.2, but in the present form, it focuses much on organic molecules, while your focus is on silica. If you wish to use this in the presentation, you could consider highlighting all the processes you studied and lower the importance of organics, or make the figure with silica in the center. The proposal lacks a synergetic figure showing all the process where silica plays a role, where the open questions are, and which the open questions are, and which open questions you will answer.
The figure has been changed in the, I mean, this is a question that in this detail refers to the first draft of the proposal. I think the key is that the condensation of silica in the presence of a reduced environment with lots of organic compounds creates biomimetic materials that are very worthy of studying, because the environment in which life very likely formed in the Hadean had a lot of these materials. And we know that this condensation of silica in the presence of organic material was happening. So clearly, this needs to be studied experimentally.
How will your project impact the field?
I think that our project will impact dramatically the field. Actually, the experiment that we have already produced and the data that we already produced already have very high impact, because what we are proposing is extremely novel. So I think that products will have a huge impact on the field of LEF origin of life.
Who are your main competitors? Which other research groups are active in this field, and why is your approach better?
There are many groups active in this field. In the United States, there's an entire system of astrobiology institutes, for instance. In Europe, there are several origins centers in Germany, in Holland, in France. But all these centers are actually focusing on very specific aspects of the origin of life, or very specific aspects of detection of life. So as far as we know, there is no center or research group that is focusing on a globally important process. And in that sense, we are sort of unique at this moment by suggesting that there has actually been a factor that operated on a global surface scale that could have triggered the catalysis of prebiotic chemistry and could have actually have been involved in self-organizational processes.
What do you think about the sulfur world scenario of the Deep, Wachter-Hauser, Russell, and many others?
These are all very interesting hypotheses. And of course, here also actually coming back to my answer of the previous question, these are all plausible hypotheses, but they operate in very specific environments. So on the surface of sulfur, or on the surface of sulfides or clay minerals, in hydrothermal systems, there are many hypotheses that are very interesting and create steps towards life. But you need very specific environments that might be not operating on a global level. What we suggest is that there are several aquatic environments, from the subaerial to the very deep submarine, that all could have been operating in prebiotic chemistry, and silica was playing a central role.
And in terms of methods, how far does the Hadean simulator differ from non-planetary atmospheric simulators?
So planetary simulators are often used to study, for instance, the conditions on the surface of Mars or other planets. And in doing so, these simulators are, for instance, mimicking extremely low temperatures or atmospheres that are very that are not dense at all, and that operate with high cosmic ray fluxes, for instance. What is unique here is that we have simulators that are mimicking Hadean conditions. And in fact, our Hadean simulator is not just one simulator, it's a whole range of instruments and laboratory tools to together mimic a situation that was likely present early on our Earth.
Do you really think that you will solve with Protos the problem of the origin of life?
No, but we will very likely take very big steps towards it. Of course, we cannot imagine whether we actually solve that problem, but with this unique and important new insight on the role of silica, which was ubiquitously present, we would have created a major step towards the origin of life.
Did you consider ethical aspects in your project?
Yes, we did. And in fact, we have very few. The only aspects that would involve ethical issues are occurring in work package, the end of work package 2 and work package 3, where we carry out some field work. And of course, and we have experience with this, whenever you do field work, you have to have obtained the permits to actually take samples and to be at certain places. We have extensive experience with this. We are also using some microbial samples for some of the experiments in work package 3, but there are no ethical issues attached to these. We have also experience with this.
Which outreach activities are you planning?
So we are planning to have a website that will be the main source of information for the citizen, because we realize that this is a project that will be very much appealing for the general public. So we will facilitate the visibility with opening different accounts in Instagram, Twitter, and also a channel in YouTube. We will do some videos. We will explore non-classic outreach scheme as comic and theater performances that we already have performed this in the past. So we plan to write papers also in popular magazine, and we will have a person which is dedicated to the management of the media and the outreach activities.
How are you prepared for the safety problem of working with methane and hydrogen?
We already do. So we have firm safety measures in place in all the 4 labs in which experiments will be conducted. Also in Work Package 0, we anticipate that an engineer will help us synchronize and adjust experimental methods, and these measures will include safety issues.
You are already collaborating. What difference will this synergy project make for you?
Yeah, so far we are collaborating mainly on the bilateral schemes, and we are already writing a couple of articles in which participate all of us. But I mean, synergy will have actually the possibility to really perform all the activity that we had designed, that we are dreaming to perform all together. So we are really anxious to have to build the different laboratories that we have designed and to use them.
What is the difference between Protos and the hydrothermal vents hypothesis for the origin of life? In particular, what is the role of silicate in hydrothermal vents?
So if you take the hypothesis of the hydrothermal vent origin of life, this focuses a lot on the creation of compartments, so-called compartmentalization for chemical reactions, and also is focusing on strong gradients in pH and strong gradients in redox. This is a very specific environment. It's basically a black smoker type environment. The role of silica is much wider than that. But of course, in hydrothermal systems, silica also has a specific role. It will be ubiquitously present. It will be precipitating there because of the temperature drop. And as it does, all kinds of self-organization will take place, which is specifically what we want to study. So we don't know the exact role of silica in hydrothermal systems. What we do know is that silica will have a role also beyond hydrothermal systems. So we think there's a much bigger role for silica than the rather limited hypothesis for the origin of life at hydrothermal vents.
Thank you, Fermin.
Okay, these are all the main questions I have. Thank you very much to everybody.
Okay, thank you for your questions, Fermin. Now we will continue the interview with Dr. Christian Geneguay, who will make questions to the group members.
Thank you very much. Thank you for proposing this interesting project and for the nice talk. My first couple of questions are more directed towards the management of the project. And in the proposal, I noticed that Bremen and Constance do not ask for any PhD students. Why is that?
That's correct. Both PIs that you mentioned asked for 2 postdoc positions. This is owing to the fact that a broad range of expertise is needed to carry out these experiments, and also to interpret the experimental findings. Also, one of the positions in Bremen will be more modeling focused, and the complexity of the models requires a certain amount of experience that we anticipate only a postdoctoral scientist brings to the table.
Okay, which mechanisms will be used to facilitate effective collaboration and communication among the 4 laboratories?
We'll be in constant exchange. We'll be using a project management system, something like Confluence or Slack, to keep exchanging ideas and sharing experimental findings and other insights we gain. I think we'll have these sort of communications on a daily to weekly basis. We'll also have a 1 week yearly meeting where all involved spend a week in a closed environment, and really that will really facilitate an intense exchange of ideas and planning of future experiments. So I think we'll be doing well on the communication side. We are all, we can't wait to start this project and interact with each other because we share this passion for silica and the magic it can do in a geological system.
Exactly.
6 years is a very long time. How will you ensure that the group stays together over the next 6 years? And how will you handle any potential conflicts between the PIs?
As Wolfgang say, we're very much anxious. So we know each other very much. We are cooperated together in the past, and we already have the task and responsibility of each of the different teams in the project clearly set. Okay. So in addition, we have a work package 0, which is devoted to the organization of the project. It will be managed by PhD scientists who has expertise in large projects at the synergy scale. So we don't see any kind of problem in the management of the project, and we cannot see any conflict. We are so much interested in making with Proto our real project, in cooperation.
And how do you plan to recruit your team members? Where will you find these specialists?
So we have a wide network of people, the top-notch groups in the field of geochemistry and geology, and also in geobiology. We think that it is not a problem. Actually, we already have people who are interested in collaborating and incorporating in products.
And in this regard, how will you ensure that you have a diverse research team?
Well, as I said, in the work package 0, there is the manager of this work package 0 will have an evaluation process to recruit a student and postdoc and technician and senior scientist. So this process, because it's unique, it's not that each one of us will recruit the people that we like it. I mean, this is something that will be, the recruitment will be performed at the project scale by the chair of the work package 0.
And if you can, how do you assure that this project will be successful?
Which one?
I think what we'll do is early on, we'll meet and talk about milestones we want to reach together. And I think the first projects we'll do will focus on strengthening the links between the different work packages. And I think that will be a good recipe to kind of have the group gel as a team. We've shown that we can work together, but these teams were, between 2 or 3 members of the group. But as a 4 PI team, we are confident that we can bring this project to fruitful results.
And if I can add to what Wolfgang just said, I think we all are fully aware that we completely need each other's research. There's a lot of interaction between our groups, and we have very much thought about how the results of one work package will feed into the other work package. So we are fully aware that we will be especially successful if our synergy is constantly maintained.
What will the field trips add?
So there are 3 field trips planned, one to Ronda region in Spain to look at ultramafic rocks, one in the Agua del Ney region in California, which has very high pH fluids, and then in Kenya, lakes that contain chert deposits. And all 3 of these localities will be important for sampling materials that are necessary for some of the experiments. So high pH fluids will be used in water-rock interaction. Ultramafic rocks will be used for water-rock interaction, and the cherts, these are modern cherts, relatively modern cherts in Kenya, will be studied with respect to precipitation processes. So these 3 field works are rather critically important. There are a lot of other samples, Archean chert samples, that we could take in ancient environments like in Australia or South Africa, but we already have a very extensive sample set. This is derived from, I have my personal collection of chert samples. So we wanted to limit the amount of field work we do because, of course, it's a large CO2 footprint we're creating if a lot of researchers need to travel all over the world.
We know that the solutions were very likely iron-rich during the Hadean. Should iron-silica interactions not be more explicitly resolved in your experimental setups? Why do you focus only on silica and not in iron?
We thought about that in great detail because indeed iron, first of all, was also ubiquitously present with silica in the ancient oceans. And also iron is, of course, a very interesting element for all kinds of catalytic reactions. We deliberately did not make a major role for iron because together with silica, we're going to greatly complicate many of our experiments. We wanted to focus on the role of silica, and therefore we have a relatively small role of iron. It specifically occurs in work package 2, where these silica iron biomorphs are going to be studied. But of course, based on all the research we're going to do in the coming 6 years, the interesting necessary next step would be to see how silica interacts with other elements, particularly iron. I totally agree with that.
Why would the development of life be less likely on other surfaces with other minerals or in another atmosphere?
They're not less likely. Actually, there are several hypotheses that suggest that these kind of processes occurred on, for instance, clay surfaces, clay mineral surfaces, on pyrite surfaces, for instance, on all kinds of minerals. They're not less likely. But these are not processes that would occur on a global scale and not in every environment. So although we do think that all these processes occur, we think that silica has a much more global impact on catalysis and on self-organizational processes.
Which is the evidence for the affirmation that amorphous silica is a catalyst?
Well, there are several papers in which it has been demonstrated that silica and silicate mineral are catalyzing the condensation of formamide. But I think that our paper published last year in Nature Scientific Communication, proving that the Miller experiment doesn't work if we perform an experiment in an inner reactor, is something that clearly proves that silica is triggering prebiotic reaction. After our paper, there is a number of papers, even a review in which people working in other fields also contributes with data about the role of the borosilicate reactor in chemical reactions.
Would you be surprised if you find in Miller-type experiments products other than amino acids?
Actually, we did, and also many other people did. Yeah.
Okay. What kind of organic, inorganic microstructures do you wish to explore?
Well, our approach is mainly experimental. So what we would like to do is to find what kind of mineral organic structure we can find in our experiment. So we are very much interested in the interaction between silica in solution and organic compound during the formation, during the prebiotic chemistry experiment, but also we will add to the reactor silicate, particularly clays, other mineral in order to trigger, because we are almost sure that we will have fantastic results in this interaction under the condition of the experiment, under the condition of the predicted for the Hadean atmosphere, this interaction between the forming organic matter, from the reducing atmosphere or the neutral atmosphere and the silica minerals and silicate minerals.
And why do you think that water condensed so early? How the zircons inform of the presence of water in the early Hadean?
So several zircon crystals have been found that are much older than the rock they are found in. The oldest is actually 4.4 billion years old. And we can study the isotopic ratio of oxygen in zircon crystals. And those oxygen isotopic anomalies show that these crystals formed in the presence of liquid water. So there is direct evidence, also because these zircons can be dated with radiometric isotopic analysis. We have a date on these zircons and we have an oxygen isotope ratio that suggests there was liquid water. This is why we have this sort of point at 4.4. There's also many zircons at 4.2 and at 4 billion years ago. They all suggest that there was liquid water present.
Thank you, Christian. We will now continue the interview with questions from Professor Enrique Pérez.
Yes, thank you for your project and your presentation. I have a few questions. Why were the oceans alkaline? On which evidence is this based?
It's not based on any kind of evidence, but it's based on what we know about how the planet works. When you look at modern Earth and the ocean composition, we know the ocean composition is controlled by rock weathering and hydrothermal reactions in the seafloor. Why should it have been different in the first 500 million years of Earth history? We know the first type of rocks were ultramafic, and Romain has talked about these reactions. And we know this from modern systems of that sort, that the fluids that are produced in these interactions are alkaline and they are reduced. And hence, there's no reason to believe that the earliest seas on the planet were not of that type, alkaline and reduced.
Okay, how will you face the problem of chirality?
Actually, this is not main topic in our project. However, we are already performing an experiment. We are performing an experiment with Teflon reactors, lining experiment, in which we use silica and single crystal of silica left side or right side. And then we are studying if there is an induction of the chirality to the amino acid and nuclear base forming. We are still waiting for the experiment results.
You focus on pH, but was redox potential more important than pH?
We think they were clearly both equally important. And in our experiments, we let basically rocks and fluids interact with each other and dial in specific ranges of pH and EH values that these type of systems can produce. and those are the pH values that likely governed the conditions in the early Earth environments. So we are not really focusing on specific ranges of pH. We look at the and pH ranges that rocks and water will produce if they interact with each other.
The soda ocean hypothesis, often linked to Kempe and Degens, is more than 35 years old. And the idea has roots, or is relevant for life sciences as well. However, it's not generally accepted. In fact, it is countered by Heilbach and Bahan. What is the difference with your approach?
Yeah, the early soda ocean hypothesis also, was considering the earliest ocean to be alkaline, but for different reasons. The idea there was that alkaline lakes, like in the East African graben systems, are good model systems for an early ocean. So alteration of ash from alkaline volcanic rocks can make water alkaline. Much more abundant were these ultramafic rocks on early Earth, and they also make waters that interact with them alkaline. So it's a different source of alkalinity. And these apparently contradicting evidences, we don't really see that much of a contradiction there, because we are talking about the Hadean, from which we don't have a rock record. These early Earth climate records that you mentioned, or the models by people like Halevi and Bahan, they concern the Archean. So that's much later. And a lot has happened, with the planet in these hundreds of million years between the origin of life and the earliest climate records.
How was the amount of felsic rocks in the Hadean and in the early Archean? Were there small continent islands, and any suggestion of how many? For example, was it a water wall full of volcanic islands?
Yes, actually, so there's many unknowns about all this simply because we don't have a rock record. It's all hypothesis, but it's generally agreed we didn't have continents, for instance, or we didn't have large cratons made of felsic rocks. So the relief on the planet, the relief basically was created mostly by impacts. So we had oceans, or we had a much shallower ocean, particularly on places where there was height differences caused by impacts and volcanism. So indeed, as you say, an early ocean might have been shallower, with here and there a volcano. Whether the entire surface was ultramafic or whether there was a significant amount of felsic rocks already, this is hypothesis. There's no consensus on this, but it is very likely that there were already felsic rocks that were, or siliceous rocks basically, that were the result of differentiation of magmas. There are several hypotheses about this. So of course, this is a transient process from the early Hadean to the late Hadean. And of course, at the end of the Hadean, we know that there were very strongly siliceous rocks, including granitic rocks.
Will your work generate new insights on the composition and variability of the Hadean atmosphere? Would you produce scenarios showing how varying methane carbon dioxide ratios will influence silicate formation or vice versa? With those scenarios, would you be able to further constrain the Hadean Earth and atmosphere?
I think we can answer this question with a yes. There's mounting evidence that CO2 and methane were both abundant in the early Earth atmosphere and oceans. So we also know that there was probably a lot of carbon you know, the general content of carbon was a lot higher than it is today, where we have all these carbonates. So yes, we will do experiments with atmospheres that have variable methane and CO2 ratios, and we'll see what organic compounds can form in these type of, you know, in a water that is in exchange with these type of atmospheric compositions. And it is very likely that this will create novel insights into, you know, possible pathways that, you know, allowed the planet to turn inhabited.
Do you have any plans to investigate in more detail the volatile and kerogenous components of carbonaceous chondrites?
These components have been exhaustively studied by many research groups, and there's a vast literature on different components that have been found in the organic phases of meteorites. We don't intend to add to that and make more analysis of these materials. What we do want to do is directly use many of these materials in our experiments, specifically in work package 3, where we will solidify these materials and see how they break down during diagenesis. And in order to do that, we of course are closely following the literature on all these materials.
I have not fully understood the importance of silica-rich oceans for the formation of protocells. How important is here also the contamination of organic matter from meteorites and other extraterrestrial bodies?
Yes, this is very important. And in fact, the role of silica can have many roles. And one of them is, for instance, acting as a protocell, simply because it creates a compartmentalization. What you need and what a membrane of a cell actually is, or a protocell, it's actually holding together the reactants for prebiotic chemistry. And that's exactly the role that silica can play as well. If you put organic materials in a silica gel, the silica gel itself might cause little pockets where these chemical reactants can react to more complex materials. So it's mostly fulfilling the same function as proto cells of organic materials.
Why do you plan to study the absorption kinetics of amino acids on silica biomorphs? Why amino acids? Are they the logical next step after formamide?
Yeah, the reason behind this experiment is because we are looking for selectivity in the absorption of amino acid by the biomorph, in order to compare the available amino acid in the solution, remaining in the solution, with the amino acid forming the first protein, the oldest protein. This is one of the reasons. And the second reason is related with a previous question about chirality. The idea also is to know if the biomorph are able to select chiral structure.
Why is it important that tungsten is tetravalent?
Yeah, this refers to one of the experiments we have in mind when we cook these ultramafic rocks with water. We talked about the fact that these systems turn very reducing very efficiently, and the conditions could be such reducing that that tungsten is tetravalent and not hexavalent, which it is usually in the environment. So we'd like to test this and see what the isotope effect associated with the tungsten redox chemistry that potentially goes on in these ultramafic hosted hydrothermal systems is. And that, you know, if we are successful, we could hopefully turn this into a proxy for reducing conditions.
what is the role of computational studies in the project? What will you model? How will you do it? How much time will you spend on this? And who will do the modeling?
Yeah, I think we'll use different types of models. I think all 4 PIs that do experiments will use some basic thermodynamic computations when they design and interpret the experiments. And then in work package 1, we have a project involved in which coupled reactive transport modeling will be conducted. We have these flow-through experimental apparatus, which we can use to generate experimental data. We can then have those experimental results inform the models. And these type of models will also allow us to do the upscaling from these, meso scale experiments to macro scale processes that we can then use to examine what likely was going on in regional environments, if not global scale.
I will add that we are also using in our diffusion reacting system, exploring self-organization, some computer that we have devoted we had designed in the past, and that we will redesign for within the Protos scheme.
Which techniques do you plan to use for the molecular characterization of prebiotic chemistry experiments? Why are you not using isotopic labeling? Who will do it?
So, actually, it's not necessary to use isotopic labeling. We will do with gas chromatography coupled to mass spectroscopy. These are techniques that we are available in our laboratories, in 2 of our laboratories. But the analysis of the result is very tricky, and it is very important to have the right libraries. And this is why we are working since about 6 years with the team of Professor Rafael Saladino in the University of Viterbo, and he will supervise all the analysis that we will do on the characterization of the organic compounds.
Why are you focusing on formamide?
Well, because formamide is the one single carbon molecule known to experience a self-catalytic condensation, forming many molecules which are very important for origin of life. Amino acid, nucleobases, PA, many other ones that are very important. And there are already many experimental results publishing and proving the formation of that silica is catalyzing the condensation of these compounds. I think that it can be demonstrated that in the cyanhydric, which is the parallel route, used by other colleagues, even in the cyanhydric route, formamide formation is very important.
You often focus on FTT. How large and far-reaching is this type of reactions? How much do they condition the evolution to extant organisms?
We use FTT synonymously with any kind of abiotic organic synthesis reaction. And there's a plethora of possible pathways. And we know from experimental work that's already been conducted, and also from empirical work in ultramafic hosted hydrothermal environments, that organic molecules are produced by reduction of carbon dioxide in these systems. And these reaction products include alkanes and alkane thiols and ketones and volatile fatty acids. And those type of compounds will be able to analyze in the experimental run products that we'll produce in our labs.
Do you have a calendar of main events for the first billion years?
Well, we have a calendar with the main events, that everybody knows. But we are anxious to really set new dates in this calendar with our synergy project.
What do you mean by life in your system?
So, yeah, there are many definitions of life, and we want to stay away from that a little bit because it just complicates everything. Actually, there is probably a whole transition from prebiotic chemistry to systems that are half alive to something that is alive in the sense that we would call it a living system. So we want to stay away from definitions and specifically focus on the actual role of silica in all kinds of catalytic reactions that would lead to systems that we would actually identify as life.
What information on biogenicity do you expect to glean from the planned sophisticated analysis of highly graphitized organic molecules?
Yeah, we don't actually focus that much on graphitized materials. This will be one of the end member results from some of the experiments in work package 3. What we actually focus on is how various organic molecules, prebiotic molecules, living systems such as microorganisms, as well as real true abiologic materials, how they actually preserve when they are embedded in silica and then are altered during diagenesis and hydrothermal alteration. But when we ramp up the temperature, and by doing so we mimic ultimately metamorphic processes, we see how these interesting characteristics of these molecules are gradually being lost, actually. So we don't expect to glean much from completely graphitized materials. What we show is what it will end up looking like if you want to compare this to highly metamorphosed ancient rocks.
Is there a link between silicon-rich early oceans and the enzymatic capability of cyanobacteria, foraminifera, sponges, and other early organisms to deposit the silicon?
Yeah, that's an interesting question. I think it's actually not known, but it's intriguing, of course. We could think about that. If silica was so ubiquitously present everywhere in the Hadean, but also in the Archean and Proterozoic, all the way up to the moment when we have organisms that use silica, such as sponges and diatoms. Of course, this is an interesting question then. Is it actually an important step in evolution? This is something that might end up being proven, actually, that silica also influenced important evolutionary steps, but we don't know that at the moment.
Do you think that your view for the early Earth can be extended to other worlds?
Yes, most definitely. And this is actually what really gets us excited about this project. We consider planetary conditions on Earth. We have a rocky part of the planet and a liquid part of the planet. All the other planetary bodies that we, look at in our solar system, Mars, not now, but 4 billion years ago, there was liquid water. You look at Titan, Ganymede, Europa, Enceladus. These are all rocky planetary bodies that have liquid water. So whatever we find in our experiments, will be valid for Hadean times on Earth, but it will also be valid for, these planetary bodies and trillions of other worlds. In that respect, and Mark alluded to it before, our environments are not specific, right? We do not